By proactively managing the identified risk factors for cataracts, you may be able to reduce their onset and their progression. Some lifestyle management strategies can avoid the oxidative damage and the glycation reactions in the lens in the eye. You should quit smoking because the toxins from tobacco smoke can damage the proteins in the eye. Limit or eliminate the exposure to UV radiation from the sun. Avoid work-related exposure to X-rays and gamma radiation, and limit or reduce the consumption of alcohol. In addition to these lifestyle changes, numerous studies have revealed food-based antioxidants are useful in the treatment of cataracts. By increasing the consumption of foods rich in antioxidants and phytochemicals, like vegetables and fruits, the human body may be able to more effectively scavenge and eliminate the free radicals and the reactive oxygen species. Other dietary considerations include avoiding meats high in cholesterol and saturated fats, and instead consuming more fish that's rich in omega-3 fatty acids. Omega-3 fatty acids were shown to protect against oxidative damage caused by UV radiation in other tissues, and since the development of cataracts was causally linked to the oxidative damage in the lens, this action could represent other mechanisms by which they protect against the cataract formation or the progression. Diabetes is a well-known risk factor for cataracts, but the link between elevated blood sugar and cataracts is less appreciated in non-diabetics. Even in people without overt diabetes, elevated blood sugar causes significant damage throughout the body by increasing oxidative stress and promoting protein-destroying glycation reactions, leading to a number of chronic diseases. The lens of the eye is particularly susceptible to damage associated with elevated sugar. Researchers at Harvard University conducted a meticulous analysis on more than 87,000 individuals over a 16-year period and concluded that the cataract may be mediated in part by glucose intolerance and insulin resistance, even in the absence of clinical diabetes. Several subsequent studies corroborated these findings. In an analysis of nearly 3,600 people aged 49 or older, the fasting glucose levels above 108 milligrams per deciliter were associated with a 79% greater risk of cortical cataract development over a 10-year period compared to concentrations below 108 milligrams per deciliter. Moreover, for each 18 milligrams per deciliter increase above the level, the risk of the progression of some type of cataracts increased by up to 25%. In a similarly designed study on more than 2,300 people, fasting glucose levels above 108 milligrams per deciliter were associated with a 2.2-fold higher risk of cortical cataract over a 5-year period. Another analysis of 3,654 elderly subjects in Australia showed that glucose concentrations between 108 and 126 milligrams per deciliter were predictive of double the risk of cortical cataracts over a 10-year period. Interventions associated with improved glucose control have been shown to reduce cataract risk. For example, in an animal model of cataracts, calorie restriction, that is, the reduction of calorie intake to a level short of malnutrition, was associated with a 27% reduction in glucose levels, fewer incidents of cataracts, and less cataract progression. Other animal studies have shown the use of anti-diabetic drug, acrobose, which inhibits carbohydrate absorption and suppresses glucose concentration, but reduces the incidence and lessens the progression of cataracts. The dangers posed by impaired fasting glucose concentrations are sadly often underappreciated by the medical establishment. Conventional physicians, in many cases, fail to take preventative action until clinical diabetes manifests, which is defined as fasting blood sugar levels of 126 milligrams per deciliter or higher. However, it's suggested to strive for an optimal fasting blood sugar of 70 to 85 milligrams per deciliter. Since glutathione is present in very high concentration in the lens and it's essential for lens transparency, it's an important endogenous antioxidant molecule in the lens. Glutathione directly scavenges the reactive oxygen species and the free radicals, preventing the oxidation of structural proteins in the lens. This is particularly important in several types of cataracts, where oxidative damage to the lens protein appears to play a key role. After scavenging reactive oxygen species and free radicals, the oxidized form of glutathione is readily recycled by a specialized enzyme. As we age, the recycling of glutathione decreases progressively. The pool of reduced glutathione decreases, and oxidized forms of glutathione begin to build up. This is especially true in the center of the adult lens, known as the nucleus, where even small amounts of UV exposure can drive free radical formation and generation of cataracts. Nutrients like N-acetylcysteine, lipoic acid, melatonin, and selenium all naturally increase the glutathione activity in the body. Vitamin C, also known as ascorbic acid, provides extra antioxidant support in the lens by acting as a sink for reactive oxygen species and free radicals. Its oxidized form, dihydroascorbic acid, is then converted back to ascorbic acid by glutathione, and indirectly in reactions that depend on glutathione. Therefore, vitamin C and glutathione are thought to work together to promote proper water balance within the lens and to prevent protein clumping. The importance of vitamin C for health of the eye is supported by the finding that vitamin C concentration in the lens are 20 to 30 times higher than those in the plasma. In addition, its importance is supported by experiments showing that inhibitors of the enzyme that recycles glutathione caused a marked increase in dihydroascorbic acid, and that the dihydroascorbic acid can cause a loss of transparency in the lens in animals if it's not converted back into vitamin C. A study examining the effect of long-term dietary supplementation with vitamin C in women showed supplementation over 10 years significantly decreased the incidence of early age-related cataracts at any location in the lens. Importantly, the study excluded women who'd previously been diagnosed with cataracts to make sure that those who started with the vitamin C supplementation at the recommendation of their doctors and as a result of their diagnosis were not included. Another study, which included a large number of men and women, revealed that high consumption of vitamin C alone, or together with other antioxidants like vitamin E, beta-carotene, and zinc, protected against the development of nuclear cataracts. A study that enrolled over 4,000 participants reported for every 1 milligram per deciliter increase in vitamin C in the serum, there was a 26% decrease in cataracts. Additionally, in a study in India that enrolled over 5,600 individuals, a strong association was found between high serum vitamin C levels and low incidences of cataract. Also known as riboflavin, vitamin B2 is a vital component of flavin adenine dinucleotide, or FAD, and this is directly involved in breaking down carbohydrates and lipids. It's important for the proper cellular energy balance, and it's also used by enzymes that recycle glutathione back into its bioactive form. It was shown in several animal models that a deficiency in dietary riboflavin can lead to cataracts. Several studies in humans have reported that riboflavin is important in preventing the formation of cataracts. In one study, women with the highest dietary riboflavin intake, as compared to those with the lowest intake, had a lower risk of cataracts. Another study reported individuals with the highest dietary riboflavin intake had an approximate 50% lower risk of developing cataracts. Vitamin E naturally occurs in 8 different chemical forms, including alpha-tocopherol and gamma-tocopherol. It possesses distinct antioxidant properties, and it prevents the accumulation of free radicals produced during fatty acid breakdown. Since it is fat-soluble, vitamin E protects fatty tissues and cellular membranes by neutralizing free radicals and reactive oxygen species. One study among women revealed that those with the highest intake of vitamin E from food and supplements had a 14% lower risk of cataracts. Another study that examined participants aged 40 to 79 years of age revealed lens opacities were more frequent in people with lower vitamin E levels. Yet another study showed the level of total tocopherol, which is the sum of the serum alpha-tocopherol and gamma-tocopherol, was associated with a decreased risk of developing cataracts. Evidence suggests the potent antioxidant lipoic acid can help to prevent diabetic cataract formation. Since it's distributed in fat-soluble and water-soluble areas of the cells and the tissues, the lipoic acid neutralizes a variety of free radicals. Lipoic acid exists in two forms, R-lipoic acid and S-lipoic acid. Of these two, the R-lipoic acid, in isolation, was shown to prevent cataract formation. The higher efficacy of the R-lipoic acid when compared to alpha-lipoic acid, which is a mixture of the two forms, might be related to a higher rate of R-lipoic acid absorption by the lens. N-acetylcysteine is a powerful antioxidant and a derivative of the amino acid cysteine, and it's been shown to prevent the opacification in the lens. It also supports the production of glutathione. By combining it with diallyl disulfide, a major organosulfide found in garlic oil, researchers have discovered that the latter could boost the antioxidant property of the former, and the combination prevented the formation of cataracts. In animal models, N-acetylcysteine has been shown to prevent the lens opacification and to inhibit cataract formation. Another study found the combination of N-acetylcysteine and glutathione ethyl ester administered as eye drops slightly inhibited the progression of diabetic cataracts at the early stage in rats. Melatonin is a naturally occurring hormone, and it's been shown to reduce oxidative stress in the lens and protect against cataract formation. Although the mechanism involved has yet to be determined, the increased production of glutathione or direct scavenging of free radicals is thought to be involved. Given that melatonin levels decline as we age, and in light of the rising incidence of cataracts in the latter part of life, melatonin supplementation may be useful for cataract prevention among aging individuals. Carnosine, along with one of its derivatives, N-acetylcarnosine, is a potent inhibitor of glycation reactions and oxidative damage. It's been shown to efficiently penetrate the lens. Like glutathione, carnosine levels decrease with age. At moderately high concentrations, carnosine was able to attenuate UV-induced aggregation of lens proteins. When delivered topically as eye drops twice per day, a solution of N-acetylcarnosine has been shown to delay the onset of diabetes-induced cataracts. N-acetylcarnosine eye drops have been shown to be effective in dogs. Visual improvements were reported in 80% of the participating dogs administered the eye drops containing N-acetylcarnosine. Taken together with information from human trials and experiments in rodents, it appears N-acetylcarnosine eye drops might offer considerable protection against the formation and the progression of cataracts. Carnitine is a natural occurring amino acid-like compound, and it's found in all mammals with essential roles in normal function of the mitochondria, the energy powerhouse of the cells. Its derivative, acetyl-L-carnitine, is a powerful antioxidant, and it's been shown to combat glycation reactions. An examination of human cataract lenses showed that as the opacification increased, carnitine concentrations decreased, with lenses containing the greatest opacification having about 30% lower carnitine concentration than those with the least opacification. In an animal model of cataracts, acetyl-L-carnitine strongly inhibited chemical-induced cataractogenesis. The researchers attributed the effects of acetyl-L-carnitine to its role as an antioxidant within the lens. In a subsequent study, researchers from the same group showed acetyl-L-carnitine also guards against self-destruction or apoptosis of the lens cell. Evidence also indicates acetyl-L-carnitine protects against cataract development subsequent to ionizing radiation exposure by upregulating intrinsic antioxidant defense mechanisms. Bioflavonoids are a class of plant-derived molecules with antioxidant properties. They may be beneficial in cataracts by helping to combat the accumulation of water within the lens cell, which disrupts the normal light refraction. Specifically, the bioflavonoid quercetin, the most widely consumed flavonoid in the human diet, was shown to inhibit diabetic cataract development, possibly acting on multiple pathways and maintaining the transparency of the lens in response to oxidative stress. Another animal experiment showed that quercetin helped to maintain lens transparency by balancing calcium, sodium, and potassium ions within the lens. Vitamin B6 or pyridoxine is an important water-soluble cofactor and is necessary for the metabolism of amino acids and the synthesis of nucleic acids required for DNA replication and repair. It's been shown to significantly reduce the production of advanced glycation end products in the diabetic lens. Though human studies in cataract patients still need to be conducted, a trial in which vitamin B6 and B1 were administered to diabetic patients showed the combination significantly inhibited DNA glycation in the white blood cells, suggesting systemic benefits. Carotenoids are a type of pigment found in plants. They absorb light and safeguard against the oxidative effects of UV rays. Several carotenoids, including lutein, zeaxanthin, and mesozeaxanthin, are not only present at high levels in the retina, but also they help to prevent cataract formation and macular degeneration. A study on a large group of women, aged 45 to 71, revealed that dietary lutein and zeaxanthin and foods rich in these carotenoids reduced the risk of cataracts that were sufficiently severe to require surgery. Another large study that enrolled people greater than 40 years of age found that those with a high dietary intake of lutein and zeaxanthin had a lower risk for nuclear cataracts. In another study on 1,802 women who were subjects in the highest quintile, the 1/5 of the distribution for the blood levels of dietary intake of lutein and zeaxanthin, were 32% less likely to have nuclear cataracts compared to women in the lowest quintile. In a small, double-blind, randomized clinical trial on 17 patients with cataracts, supplementation with lutein, 15 milligrams, 3 times per week for up to 2 years was associated with improved visual acuity. Bilberry is rich in anthocyanins, which are plant pigments, and they exert a variety of beneficial effects in the human body, including functioning as antioxidants and modulating inflammatory processes. Importantly, these anti-inflammatory and antioxidant effects were observed within the eye in an animal model. In an experimental model involving animals prone to age-related eye disease, such as macular degeneration and cataracts, long-term administration of bilberry extract completely abrogated the impairments in the lens and the retina, whereas 70% of the controls developed the cataracts and macular degeneration. Bilberry has yet to be studied in large trials to assess its effects on cataracts in humans, but controlled studies have found benefits associated with bilberry supplementation, alone or in combination with other nutrients that support eye health, for eye strain and glaucoma. Green and black tea contain antioxidant molecules called catechins and polyphenols, and these have been studied in many human health conditions. Several animal studies have shown that green and or black tea can mitigate the cataract formation or progression. In one such study on rats with chemical-induced diabetes, green and black tea administered in drinking water was shown to retard the cataract development by reducing the detrimental effects of elevated glucose on the lens. Another study found that green tea extracts bolstered antioxidant defenses and reduced the incidence of chemical-induced cataracts in an animal model. A similar study on an animal model of chemical-induced cataracts revealed that green tea and black tea extracts slowed the progression of lens opacification. Resveratrol, a natural polyphenol found in several plants, including grapes, peanut, and pine, has multiple health benefits due to its function as an antioxidant. In an animal model of chemically induced cataracts, resveratrol was shown to reduce oxidative stress in the lens and suppress cataract formation. Resveratrol was also able to increase the survival of human lens epithelial cell cultures that were subjected to oxidative stress, and it decreased the cellular markers of aging. These effects could be explained by its ability to increase the activity of intrinsic antioxidant enzymes, superoxide dismutase, catalase, and heme oxygenase. Selenium is a trace mineral and it's involved in many biological functions within the body. Studies have shown that selenium can slow the development of cataracts by lowering oxidative stress in the lens. Although additional studies are required to determine how the selenium prevents oxidative damage in the lens, it's been shown to enhance recycling of glutathione. Vitamin K is a fat-soluble vitamin and it's found in leafy greens like spinach and collard greens. Phytoquinone, or vitamin K1, is the primary source of vitamin K in the diet. Vitamin K1 has been shown to inhibit the enzyme aldose reductase, which converts glucose into sorbitol, a sugar alcohol. Accumulation of sorbitol in the eye lens contributes to the pathogenesis of cataracts. Preventing the accumulation of sorbitol and its degradation products, fructose, in the lens decreases free radical stress and it lessens osmotic pressure, decreasing the risk of cataract formation. A retrospective analysis of a large randomized control trial found that greater dietary vitamin K1 intake was associated with lower cataract risk. The trial enrolled 5,860 Mediterranean people and it assessed dietary vitamin K1 intake via a food questionnaire. After nearly 6 years of follow-up, the participants who consumed the highest levels of vitamin K had a 29% lower risk of developing cataracts than those with the lowest intake. For more on herbs, supplements and natural treatment plans, check out my website.